Stemmedicare, noble care for human nature. Extracellular vesicle therapeutics without immune rejection. Regenerative medicines refer to treatment that replace damaged cells, tissues, and organs or restore their functions. They are attracting attention as fundamental treatments for incurable diseases such as Alzheimer's, spinal cord injury, and cancer. The most well-known regenerative therapy is cell therapy, especially stem cell therapy. This means a therapy that injects stem cells from one's own body or a healthy donor into the body for various therapeutic purposes. Autologous cell therapy. One's own stem cells or immune cells are not dangerous by themselves. However, the risk caused by animal-derived ingredients used during the cell culture process must be considered. Also, since it can only be used by the patient himself, it is inevitably very expensive. Then, what about cell therapy products using other person's cells? All cells contain antigenic molecules on their membranes that can distinguish between self and non-self. These molecules are called major histocompatibility complex MHC. In humans, they are also called human leukocyte antigen HLA. HLA types are also different between parents and children, or between siblings, and even during bone marrow or organ transplantation. HLA typing analysis is essential to determine whether transplantation is possible. Allogeneic cells administered to the patient are recognized by T cells, which are the most abundant immune cells in the patient's blood, and the T cell receptor binds to the HLA molecule of the allogeneic cells to eliminate non-autologous cells. This process is called immune rejection and is the biggest barrier to the development of allogenic cell therapy products. How about the extracellular vesicle therapy? Extracellular vesicles, EVs, are nano-sized particles limited by lipid bilayers that are released from living cells. EVs contain various bioactive substances such as proteins, mRNAs, and microRNAs, and have various therapeutic effects by delivering them to other cells or tissues. Since EVs are very small compared to cells, they are receiving great attention as an alternative to cell therapy without immune rejection. Like cells, extracellular vesicles express HLA molecules on their membrane. So, the administration of allogeneic extracellular vesicles can cause undesired immune rejection by MHC mismatching. Furthermore, the rapid proliferation of T cells is activated by MHC mismatching of the administered allogenic extracellular vesicles, which may cause allergic reactions. To reduce the risk of immune reactions by allogenic extracellular vesicles, it is recommended to match HLA typing between donors and patients, or to use immunosuppressive agents. In addition, the allogeneic extracellular vesicles are quickly internalized and degraded by the patient's macrophages. It has been reported that more than half of administered allogeneic extracellular vesicles are eliminated within 2 minutes by endocytosis of patient macrophages. Allogeneic extracellular vesicles absorbed by macrophages are mainly detected in the lungs, liver, and spleen, where many immune cells are present, and very few extracellular vesicles are delivered to other organs, including the brain. Thus, the therapeutic effect of allogeneic extracellular vesicles is also significantly reduced. Therefore, it is very important to develop allogenic cell and extracellular vesicle therapeutics that do not cause immune rejection so that anyone can use them safely. So, how can you overcome immune rejection? We found the key to solving immune rejection during pregnancy. How can a fetus with a different HLA typing from the mother be protected from the maternal immune system during pregnancy? The key to protecting the fetus from the mother was a protein called HLA-G, which is produced only during pregnancy. The implanted embryo and subsequent fetuses are protected by trophoblast cells produced only during pregnancy. Trophoblast cells express and secrete HLA-G protein by the stimulation of progesterone, a pregnancy hormone. An immunotolerant environment that protects the fetus from maternal immune cells is established by HLA-G. HLA-G preferentially binds to the inhibitory receptors of maternal immune cells, including T cells, NK cells, B cells, dendritic cells, and macrophage, over other HLA molecules, leading to induce on immunotolerant environment that protects the embryo and the fetus from them. HLA-G bound to inhibitory receptors of immune cells reduces immune responses such as cell proliferation and cytotoxicity reduction, production and secretion of inflammatory cytokines, and phenotypic changes to immune tolerance characteristics. As such, a phenomenon in which the immune response of immune cells is reduced by binding to cells expressing HLA-G is called local immune tolerance. Unlike other HLA molecules, HLA-G has both membrane-bound isoforms and soluble isoforms. HLA, G1, 2, 3, and 4 are membrane-bound isoforms, and HLA-G5, 6, and 7 are soluble isoforms. In particular, HLA-G secreted from trophoblast cells combines with a wide range of various immune cells to establish a broad immune-tolerant environment without direct cell contact. This phenomenon is called extensive immune tolerance. Extensive immune tolerance can also be established by HLA-G containing extracellular vesicles. Extracellular vesicles secreted from tolerized cells that can express and secrete. HLA-G can also induce a broad range of immune tolerance environments by binding to inhibitory receptors on various immune cells, thereby preventing EV-induced immune rejection. How can we establish immune-tolerogenic cells expressing and secreting HLA-G as allogeneic cell therapeutics without immune rejection due to mismatching MHC? We developed an ex vivo culture system that simulates in vivo environment during pregnancy, in which the expression and secretion of HLA-G are induced only in trophoblasts. We also demonstrated that it is possible to establish immune-tolerized cell lines capable of expressing and secreting HLA-G in cells other than trophoblasts. Each HLA-G protein moves to the cell membrane, then membrane-bound HLA-G proteins are expressed on the cell membrane, and soluble HLA-G proteins are secreted out of the cell. Through this process, an immune-tolerized cell line having characteristics capable of expressing and secreting HLA-G proteins is induced. Extracellular vesicles secreted from immune-tolerized cells capable of expressing and secreting HLA-G also express HLA-G molecules together with MHC molecules. So immune-tolerized extracellular vesicles that do not cause immune rejection due to MHC mismatch can be established. We have established the world's first ex vivo cultivation platform technology for inducing immune-tolerized HLA-G positive cell lines by simulating pregnant environments. In the case of stem cell therapy, which in the mainstream of regenerative medicine, it is possible to develop allogeneic stem cell therapy that can be safely applied without immune rejection. In the case of hematopoietic stem cell transplantation, anyone can safely transplant donated hematopoietic stem cells from a healthy person without having to wait for years to find a suitable donor. In addition, in the case of CAR-T or CAR-NK therapeutics, which are currently considered mainstream immunotherapy, the scope of application can be expanded to allogeneic therapeutics beyond the limits of patient-specific treatment. In addition, in the case of organ transplantation such as liver, kidney, heart, etc., immunosuppressants must be used for a long period of time, but by applying the present platform technology, organ transplantation can be performed safely without using immunosuppressive agents that cause numerous side effects. Immune-tolerized extracellular vesicle therapy. On the other hand, since it has been proved that extracellular vesicles secreted from immune-tolerized cells that express and secrete HLA-G also contain HLA-G, it is possible to develop an allogeneic extracellular vesicle treatment that can be safely used by anyone without immune rejection. Unlike other allogeneic extracellular vesicle therapy products with immunogenicity, which are immediately internalized in macrophages and found only in the liver, lungs, and spleen, HLA-G containing extracellular vesicles can be delivered throughout the body without immune rejection, so it can be developed as a next-generation regenerative therapy. In particular, extracellular vesicle therapeutics can pass through the brain-blood barrier, which cell therapeutics cannot pass through and can be delivered to damaged brain and nervous tissue, so they can be developed as treatments for various neurodegenerative diseases such as Alzheimer's and Parkinson's disease. By overcoming immune rejection, Stemmedicare has established platform technology for developing cell and extracellular vesicle pharmaceuticals that anyone can safely use regardless of time and place.